Welcome back to my course Industrial Biotechnology. Now in the last class I shared with you the wine and vinegar fermentation process. I told you that wine, there are different types of wine that has been marketed, and broadly it can be classified into two types. One is called still wine, another is called sparkling wine. Still wine doesn't have any kind of carbon dioxide, but still wine we have carbon dioxide. It may be done by using carbonation or by natural fermentation process. Then also that wine can be divided into other different category that has been explained, but mostly the wine produced from the grape, and grape should be sound and ripe. Sound means I told you that texture of the grape should be hard, and if it is soft, there is a possibility of microbial contamination in the fruit. And then ripe it should be there because so that the sugar content is more. Now wine fermentation process is usually carried out in two different stages. One is called primary fermentation, another is called secondary fermentation process. Main problem associated with the wine is the storing, because I told you that if you store the wine at for 2 years, we get the dry wine. Dry wine means it doesn't have sugar for taste perception. We cannot feel it, the sweetness of the wine. Then we call it dry. The sugar concentration will be as minimum as possible. And then also we have fine wine that required 5 years or more than, because during the storing I mentioned that alcohol and acid, the trace amount of acid that present in the wine, they will react with each other and form the ester. So harshness of the wine will go out, and in place of that some good flavor will be developed, because we know the different esters are responsible for developing the flavor. Now again, wine has two type of marketing. One is short time and long time. In the short time market, we just use some kind of preservative and do it in the market, but in a long time we do the pasteurization of wine, and so that it can be used for longer period of time. And wine is used, or alcohol is used for the production of vinegar. I told you vinegar is a Latin word, and this is nothing, this is something, this is called sour wine. Sour wine means the wine when in presence of some acetic acid bacteria. The example is the acetic acid, Acetobacter aceti, that convert ethanol to acetic acid. So now concentration of acetic acid varies from 4 to 20%. The different type of vinegars are available. Cidered vinegars is quite common in the market, that is usually produced from apple juice. Apple juice undergo the alcohol fermentation process followed by the acetic acid fermentation process. Now we have, I try to point out the difference between the natural vinegar and the synthetic vinegar. In case of synthetic vinegar, we use the pure acetic acid for the preparation of the vinegar, and this is available in two different forms. One is colorless, another is colored. In case of color, we add some kind of caramel. Caramel is nothing but when you heat the sugar undergo caramelization reaction, it gives a black color. We make a solution in water, and that gives a color that is a permissible color as per Food Product Organization is concerned. And then this is industry, it is produced in two ways. One is French process, another is German process. I told you the French process is quite old process, but we get good quality vinegar out of this. But German process is usually a little bit faster as compared to this French process. Now today I am very happy to share with you another fermentation process, that is citric acid. And citric acid is such a wonderful product that has been used in different for different purposes can be used. Citric acid, if you look at citric acid, is considered as a tricarboxylic acid, though it has three carboxylic acid groups that we have, and it has the property like this. It is available in the two different forms. One is called anhydrous citric acid, another is citric acid monomer. Citric acid monomer, this formula is a little bit wrong, you please correct it. This should be H8, because this is 6H8O7, this also should be H8. So this is citric acid monomer. This molecular weight is 100. The citric acid anhydrous has the molecular weight 192, and this citric acid monohydrate, that is 210, and specific gravity more or less they are close to each other. Melting point 153 degrees centigrade in a citric acid anhydrous, and in case of CAM it is loses water at 70 to 75 degrees centigrade. Most important thing I want to point out here, the transition temperature is 36.6 degrees centigrade, because this anhydrous and monohydrate, because when you do the crystallization, if we keep the temperature below 36.6 degrees centigrade, we get citric acid monohydrate, and if we keep the temperature higher than that, we produce the citric acid anhydrates. Now this is the stoichiometry of citric acid production. This is how your sugar, it produces the 2 moles of this sugar is comprised of glucose and fructose. It is 2 moles of 6,6H12O6, and then this produces 2 moles of the CAM, what we call citric acid monomer. Now if you see the yield, CAM yield, if it is calculated, is about 123%, and citric acid anhydrous, if you calculate, here I want to draw your attention that yield, that it may be more than 100% also. Now here I want to just point out, in case of ethanol fermentation process, we know that 180 gram glucose produces 92 gram of ethanol. So there the yield is about 51%, around 51%. But here in case of SCAM, it is here you see that 123%. So yield may be less than 100%, more than 100%. It depends on the type of product we are producing. So that is thing. And if I can give another example, that Bacillus coagulans, it has the glucose isomerase enzyme that can convert glucose to fructose. Now glucose has the same molecular weight as that of the fructose, so there the conversion is 100%. So now let me talk about, I told you that the citric acid has tremendous potentiality in the market. It has different use, and I work with Citrugia Biochemicals, which is located in Surat, and this is was in collaboration with John and Starch Limited, UK. That now use of citric acid. Citric acid is a weak organic tricarboxylic acid having the empirical formula this C6H8O7. 75% citric acid is used in food, confectionery and beverages. So most of the citric acid, because not only it is used for acidifying the product, but it gives a typical flavor, and 10% is used for pharmaceutical industries and 15% used by the other industries. So 10% the pharmaceutical industry, mostly they use the citric acid anhydrous, and in 75% citric acid and 50% that industrial use, mostly we have CAM. Now it is used as a food acidulant, because lot of jam, jelly, orange squash, all places we use this citric acid. It has a pleasant taste and enhance the flavor. It is the most important thing is it is a weak acid. It can be used for cleaning the power station boiler, because we know during the steam generation, there is the deposition of the solid material on the boiler tubes that is usually cleaned by the citric acid. But if we use the mineral acid like HCl, H2SO4, that may affect the material of construction. So it is recommended that we should use the weak acid for cleaning the scale formation in the boiler tubes. Now, besides that, the citric acid at other uses like ferric ammonium citrate tablet used for the anemia patient. Also, we know that in the biotechnology lab we use the citrate buffer. Then citric acid used also for the preservation of ascorbic acid, which is called vitamin C, also for the stabilization of the fat. So it has tremendous potentiality in the market, and I can remember when I worked with Citric Biochemicals, I could not find any trace amount of product in our godown. As soon as it is produced, it is marketed, because the Citric Biochemicals was the only company in India which was producing citric acid through the fermentation process. Other company was Citrate India. They usually imported the calcium citrate from China, and you produce citric acid from calcium citrate. That is not a fermentation process, that is a chemical process. Now, if you look at the history, is very interesting. The Seeley that in 1784 first isolated the citric acid from lemon juice. We know lemon juice content is called citrus food. So citrus name is coming from the citric acid and crystallize it and as a solid. So I mentioned that two type of organic acids we have. One is called volatile fatty acid, and another is non-volatile fatty acid. Volatile fatty acid means if you keep it to the atmosphere, it will evaporate it out, and non-volatile fatty acid means if you keep it up to some time, that your water will be evaporated out and crystals of that acid will remain. So citric acid is considered as a non-volatile fatty acid. Weimar, this 1893 described the citric acid as a product of mold fermentation. Then 1917, the Curie published the results of citric acid can be produced from Aspergillus Niger. The pharmaceutical company Pfizer began the industrial level production using this technique, but 2 years later, then John E. Starch Limited UK took the technology, or purchased the technology from the Pfizer and marketed throughout the world. 2007, the worldwide annual production stood at approximately 1,600,000 tons of products. Now, different organisms can be produce citric acid. We have Aspergillus, different Aspergillus species, Aspergillus niger, Aspergillus clavatus, Penicillium luteum. But among all these species, the organism that is mostly used by the industry, that is the Aspergillus niger. Now, Aspergillus niger is the various strain was found to produce citric acid. Aspergillus niger is has the industrial importance. Now, this particular strain is available most of the rotten citrus food. If we have in the market that any kind of rotten citrus food, we will find the black spot formation is there. This black spot is nothing but Aspergillus niger. Now, if you pictorially, it looks like this. The black spots looks like this, and under microscope it looks like this. It is beautiful that looking that kind of flower type of things that we can observe. Now, the advantage of using Aspergillus niger for industrial use is that the high yield, uniform biochemical characteristics, and the strain we use in the industry, that is the industrial strain, that is not the wild strain. Naturally, that productivity of this strain is very high. So it can produce high concentration of citric acid. It can use the high concentration of sugar for the production of citric acid. Now, uniform biochemical characteristics, easy cultivation and production of negligible quantity of undesirable end product, and it looks like this. Another picture we have taken from the website. It looks like that. Then this is the spores. You can see that. Now, biochemistry of citric acid fermentation process. Citrate is the intermediate of the TCA cycle, a central metabolic pathway of animals, plants and bacteria. Particularly aerobic organisms we follow, we have all the aerobes and anaerobes, they have the endormyro pathway through which we can produce the glucose is converted to pyruvic acid, but aerobes they have mostly the TCA cycle. Through the TCA cycle we can produce carbon dioxide and water. So, it is like this. The glucose is a predominant carbon source for the citric acid. I have already showed the stoichiometry. Biosynthetic pathway of citric acid production involves the glycolysis, wherein the glucose is converted to 2 moles of pyruvic acid. The pyruvate in turn forms the acetyl CoA, and acetyl CoA in combination with oxaloacetate with condensates to give the citrate. So, it is the metabolic pathway looks like this, that we through the metabolic pathway, that endormyro pathway we produce pyruvate, and this pyruvate produce the acetyl CoA, then oxaloacetic acid. This is the one of the metabolites of the TCA cycle. When they combine together with the help of citrate synthesis, they produce the citrate. And then again if you allow the citric acid product, they allow the TCA cycle to continue, then again it produces isocitrate, alpha ketoglucarate, succinate, succinyl CoA, succinate, fumarate, the malate and the oxaloacetate. So, that this is the cycle is going on through which we produce I told you it produces carbon dioxide. We know that when we take glucose in our system in the day-to-day life, we get a lot of energy, and this energy comes from the energy molecule produced through these biochemical reactions. So, this is during this the product is ultimate product is same as if you burn glucose, you produce carbon dioxide and water. If you take in our system glucose, that also produce carbon dioxide and water, but only that the difference is that when you burn glucose it produces heat, but when you pass through this metabolic pathway we produce some kind of energy molecule that we use as per our requirement. But here we have our interest is the citric acid production. So, in the citric acid production, the beauty is that if you give the inhibition to the enzyme responsible for the formation of isocitrate or alpha ketoglutarate, that is the enzymes is aconitase and isocitrate dehydrogenase, then the accumulation of citric acid will take place. That is actually happen in case of citric acid fermentation process. When we work with this process, then we found that first 24 hours there is no accumulation of citric acid, then the TCA cycle will be in operation. But after say about 46 hours or 48 hours of fermentation, there will be accumulation of citric acid, and we find the inhibition of isocitrate dehydrogenase. And then if you continue after 96 hours of fermentation, the total activity of aconitase and isocitrate dehydrogenase will be inhibited. So your accumulation of citric acid will be greatly taking place in the system. The normal time of citric acid fermentation is 124 to 144 hours. That is approximately 6 days. Now, production of citric acid can be produced in 2 different ways. One is called surface culture, another is submerged culture. Surface culture, because this is the aerobic fermentation process. Surface culture means that we keep the more surface areas, so that natural that diffusion of oxygen that takes place in the fermentation media and growth of the organism take place. But this technique is very less used in the industry, and mostly we use submerged culture because suppose I can give the example. Suppose we have special wide mouth Erlenmeyer frogs. We know that this is like this. This is so this is wide. Here we put the media, and wide the surface is there, so the diffusion of oxygen take place at the surface of the media, the growth of the organism. If you take the side view, it will like this. This is on the surface, the microbial organism will grow on the surface of this, and in this media you may get the citric acid production. This is very slow fermentation process as compared to submerged fermentation process. In case of submerged fermentation process, what is happening, that whole media is used for the growth of the organism, because here you have starter and you put the organism, the whole, here the organism growth only in a surface culture growth take place on the surface of the media, but in case of submerged culture the growth of the media will take place throughout the media. That is the difference. So here surface culture we have solid media, we have liquid media, and submerged culture we have start reactor and airlift reactor. Though we have mentioned too, the mostly we use the start tank reactor for producing the citric acid. Now I shall discuss surface fermentation. The solid nutrient media is used, that in case then use the for the production of complex flavor extracts by bacteria and molds, that is the Aspergillus and Niger. Limited water availability, then water is placed in simple thermostat box on baking tray like place to which the microorganisms are included. So this is the surface culture that we have, and surface culture, as I pointed out, fluid nutrient media is used, are propagated in the bioreactor, sufficient water is availability is there. So here growth is more and production of citric acid also will be more. Now question comes that what are the nutrients required for this, and this is very important as far as citric acid fermentation is concerned, because if you look at this carbohydrate sources, a high yield occur if the sugar are rapidly metabolized are used, as for example sucrose, glucose and maltose, and cane molasses and beet molasses are commonly used. Now here we have little problem, because when we use the cane molasses, then cane molasses content it is also a good media for the growth of yeast cells. Now yeast cell, I already pointed out, yeast, the Saccharomyces cerevisiae, they have very low doubling time, and whereas the fungi, that Aspergillus Niger, has high doubling time. So since we know the doubling time Td equal to ln2 by mu. The mu is the specific growth rate, ln2 by mu. So it is inversely proportional. So if doubling time is less, that means if it is less, then mu will be high, and if doubling time is high, that means mu will be less. So since mu is yeast, that mu is more as compared to Aspergillus Niger, then that in the citric acid fermentation process, the main problem we face with the yeast contamination problem, because if any yeast enter into the system, then it affects the citric acid production to a great extent. Now trace element plays important role in the citric acid fermentation process. The certain trace element like iron, copper, zinc, manganese, magnesium, that cobalt are essential for the growth of the Aspergillus Niger. Manganese ion promote the glycolysis and reduce the respiration. Iron is a cofactor for the enzyme aconitase in the TCA cycle. Iron concentration 0.05 to 0.5 ppm is ideal for the optimum citric acid concentration. Now here I want to point out that citric acid concentration, that is mineral concentration, plays very important role, what we call pivotal role in the citric acid fermentation process, particularly if we consider the iron and manganese. This plays very vital role as you have seen that iron is a cofactor for aconitase, which is I have already shown you. You can see here that this is aconitase, is the citric. If this enzyme is inhibited, then and only then citric acid accumulation will take place in the system. So that is why that if iron we take out from this media, then and only then the activity of aconitase will be affected. So this is very important. So this iron and manganese that plays a very important role, and manganese that phosphofructokinase, there is the enzyme. If we reduce that this manganese concentration in the media, then citric acid inhibition for phosphofructokinase that will be reduced, because if the activity of phosphofructokinase inhibited in the Embden-Mayrov pathway, then our citric acid yield in the fermentation process will be reduced to a great extent. So this is the problem that we have. So this is we have observed that if manganese concentration is reduced by 1 ppm, is increased by 1 ppm, in other way our citric acid yield reduced by 10%. The reduction of citric acid will take place about 10%. So this is that you can see that what is tremendous things that we have effect we have on this citric acid fermentation process. So because as is pointed out that cane molasses is found most suitable substrate for this citric acid fermentation process, and it contains good amount of iron and manganese. So when the cane molasses is used for the inoculum vessel, then I shall show you that how the inoculum cell production take place. Now in the inoculum vessel, that when you use, then we allow the citric acid to continue. So we required the iron and manganese sufficient amount, so that is not a problem for the Aspergillus Niger. But when you go for this production fermenter where we are looking for citric acid production, this is to be removed. Now question comes, how we remove citric acid from the cane molasses? So we use some kind of chelating agent, potassium ferrocyanide. We steam it, we boil it for half an hour, so that whatever iron and manganese present in the cane molasses, that will be precipitated out. This is how we can remove. And this is the concentration of the media, different is given here. Then pH of the fermentation process is usually 2.5 is preferable. At low pH, transport citric acid is much higher. At pH above 4, the gluconic acid accumulation take place at the expense of citric acid. Now here I want to point out that if you maintain the control pH, that at 4, then we will having the glucose, we will not get much of citric acid production. But if you control the pH goes beyond 6, then we will get the oxalic acid production in the system. But if we allow the pH to drop down, then we will find sufficient amount of citric acid production in the system. So another advantage, at low pH, the risk of contamination is very minimal, and since many organisms cannot grow at this pH, dissolved oxygen plays important role, because dissolved oxygen, because this is the aerobic fermentation process, so aeration is very important factor for this fermentation process. Now nitrogen source, ammonium salt, nitrate and urea are used as a nitrogen source. Now here I want to point out another very interesting thing, that since this is the filamentous cells, that when you use any kind of start tank reactor and we inoculate the Aspergillus Niger, if we put on the agitator, the growth of the organism will be affected drastically. So initially there should be an idle time for both the inoculum vessel and the production fermenter, so that your organism can grow sufficiently. Then and only then we can put the agitator on, so that your organism can go in the media properly. Let me stop it here, and next class I shall continue this lecture. Thank you.